Bismillahirrahmanirrahim. Assalamu alaikum dear viewers. I am Dr. Muhammad Usman, and this is the first video about thyroid hormone, in which we will discuss the synthesis, transport and mechanism of action of thyroid hormone. Then in the second part, inshallah, we will discuss its functions, and in the third and last part we will discuss the abnormalities related to the thyroid hormone. So let's begin. Now, you know that thyroid is one of the largest endocrine glands in our body. It is located in front of the trachea, as you can see. Its weight is about 15 to 20 grams, and it receives one of the richest blood supplies in our body. It secretes three hormones, that is thyroxine, T4, triiodothyronine or T3, and the third hormone is related to the calcium metabolism, and that is called calcitonin. Now, this is the physiological anatomy of the thyroid gland. The gland is composed of large number of thyroid follicles, and each follicle contains this colloid material in its lumen, and this colloid is surrounded by these cuboidal epithelial cells, okay, thyroid cells or cuboidal epithelial cells. The main component of the colloid is a very large protein, thyroglobulin, and thyroid hormones T3 and T4 are formed here, and they are incorporated in the structure of thyroglobulin, as we will see subsequently, inshallah ta'ala. Now, these are the steps of synthesis and secretion of thyroid hormone, and the first step is iodide absorption from GIT. Okay, iodide absorption from GIT. Now, our normal iodine requirement is about 1 milligram per week, and the iodine ingested orally, this is well absorbed from the GIT. Then the next step is iodide trapping, that is how the iodide is transferred from the blood into these thyroid cuboidal epithelial cells. So, this is the follicle, picture of the follicle and thyroid cells, and this is one enlarged this cuboidal epithelial cell. Okay, we have enlarged one cell, and this is the basal side of the cell which faces towards the blood capillaries, and this is the apical side of the cell. Okay, this is the apical side of the cell, and this is the basal side of the cell. Now, on the basal side of the cell we have this transporter, and it is called NIS, sodium iodide symporter, and from here, from the blood capillaries, the iodide enters the thyroid cells by means of this sodium iodide symporter, which transfers one iodide ion and two sodium ions at the same time into the cell. Now, once the iodide is inside the cell, it is transferred to this apical side, and then another transporter which is called pendrin or chloride iodide counter transporter. It acts and it carries the iodide into this lumen, okay, lumen of the follicle. So, what happens next, that but before that I would like to tell about the role of TSH, thyroid stimulating hormone. The thyroid stimulating hormone, which is released from the anterior pituitary, it basically controls the secretion and formation of thyroid hormone by stimulating this step, that is iodide trapping. Okay, the hormone acts on this pump or this transporter and speeds up its activity. So, more and more iodide is pumped from the blood into this thyroid cell. Now, the next step is formation and secretion of the thyroglobulin. Basically, these thyroid cells, they synthesize and secrete into the lumen of the follicle a very large protein, which is actually a glycoprotein, thyroglobulin. Its molecular weight is about 3,35,000. Okay, each thyroglobulin molecule contains 70 tyrosine amino acids, and these tyrosine amino acids, they basically combine with iodide to form the thyroid hormone. Now, the next step is oxidation of the iodide ion. Now, once the iodide is here in the lumen of the follicle, it is oxidized in the presence of peroxidase and hydrogen peroxide to its oxidized form. Okay, this is the oxidized form of the iodine, and only this form is capable of combining with the tyrosine. The next step is iodination of the tyrosine or formation of the hormone. Now, in this step the iodine combines with tyrosine, and this occurs in the presence of enzyme peroxidase. So, together they form monoiodotyrosine. Then two monoiodotyrosine combine and they form diiodotyrosine. Then an MIT, that is monoiodotyrosine, and one DIT, that is diiodotyrosine, they combine in the presence of the same enzyme peroxidase to form T3. Okay, 2 from here and 1 from here. So, 2 plus 1 3. So, T3 hormone or 3,5,3 triiodothyronine hormone is formed. Okay. Then 2 DIT, they combine, one DIT and another DIT, they combine in the presence of again same peroxidase and they form T4 or tetraiodothyronine, which is the main hormone. Then the next step is storage of T3 and T4, and each once the hormone has been formed, now each thyroglobulin molecule, it contains up to 30 T4 molecules and a few T3 molecules. Okay, as you can see here, each thyroglobulin molecule Tg, it is shown here as Tg. Okay, it contains in its structure about 30 T4 molecules and a few T3 molecules. And if thyroid hormone stops, this stored form is sufficient for 2 to 3 months. So, in this way it is stored in the lumen of the follicle. The next step is secretion of hormones. Okay, secretion of hormones. Basically, it occurs by pinocytosis. Okay, from the apical side of the cell, thyroid cell, a pinocytic vesicle it projects. Okay, there is pinocytic extension, pseudopod is formed, and then it engulfs some part of the colloid, and this colloid contains thyroglobulin. So, in this way a pinocytic vesicle is formed and it comes into the this cytoplasm of the thyroid cell. Okay, a colloid droplet is formed, as you can see here. Now, lysosomes come to play their role and they combine with this colloid droplet. Okay, lysosomes, they combine with this colloid droplet on thyroglobulin, and the proteases, as you can see here, enzyme proteases, they act upon the thyroglobulin and they cause release of T3 and T4 hormones from the structure of thyroglobulin. And once the T3 and T4 hormones are formed, they are released into the circulation from the basal side of the cell, as you can see here. Okay, and RT3 stands for reverse T3. Some amount of this hormone is also formed, but it is inert and it has no physiological action. Now, we come back to the thyroglobulin. Now, from the structure of the thyroglobulin, some MIT and DIT are also released. Okay, when there is cleavage of the thyroglobulin, then along with T3 and T4 formation, some MIT and DIT are also released. Now, another enzyme, deiodinase, it acts upon this deiodinase, it acts upon MIT and DIT and it causes their deiodination. So, what do we get? We get tyrosine and iodine, and both of these are recycled. Okay, tyrosine again is incorporated into the thyroglobulin molecule, which is then secreted into this lumen of the follicle, and again iodine comes into this cycle and it is also secreted into the lumen. Now, this protein megalin, it causes endocytosis of the colloid, that is thyroglobulin. So, some amount of the this thyroglobulin, it is also endocytosed with the help of this protein megalin, and this thyroglobulin by transcytosis, it comes to this basal side of the cell, and then it is some amount of thyroglobulin is secreted into the blood. So, this is the mechanism of storage and secretion of T3 and T4 hormones. Now, we will discuss the transport of T4 and T3 hormones to the tissues. Basically 93% of the hormone which is secreted from the gland, it is thyroxine or T4, while only 7% T3 is secreted. Almost all the thyroxine or T4 is eventually converted to T3 in the target tissues by deiodinase enzyme, which are present in target tissues also. But you must remember that T3 is about 4 times more potent than T4, but it is present in the blood in much smaller quantities and persists for a much shorter time compared with T4 or thyroxine. Now, how the transport occurs, that 99% of T4 and T3 they combine with the plasma proteins. Mainly they combine with the thyroxine binding globulin, which is called TBG, thyroxine binding globulin, and some amount is combined with the thyroxine binding prealbumin, TBPA, and also with the albumin. Now, from the plasma protein pool, T4 and T3 are released slowly into the tissue cells. These hormones have slow onset, as you can see in this figure. Okay, on the x axis we have duration, days, and on the y axis we have the action of hormone in the basal metabolic rate. So, these hormones have slow onset of action and long duration of action. Now, let us talk about the mechanism of action of this hormone. This hormone acts by genomic way, it causes a genomic action, and it has also a non-genomic action. Now, in the genomic action, what happens, that as you can see in this figure, both the T3 and T4 they easily cross the cell membranes and they come into the cytoplasm. Now, here the iodinase enzyme is present and it converts T4 into the T3, and this T3, as you can see here and here, it easily crosses the nuclear membrane and it comes into the nucleus. Now, here on the genes or chromosome there are receptors for the thyroid hormone, which is called thyroid hormone receptor, and it is always present in combination with this retinoid X receptor or RXR. Now, together this all is called thyroid hormone response element. As you can see here, this is called thyroid hormone response element. Now, it causes gene transcription, and as a result of gene transcription we get messenger RNA, and then translation occurs and a lot of new proteins are formed. The net result is a generalized increase in functional activity throughout the body. So, as you can see here, this hormone has got very diverse effect. Regarding metabolism, it increases the activity of the mitochondria, it increases sodium potassium ATPase pump activity, it increases oxygen consumption, then glucose absorption, gluconeogenesis, glycogenolysis, lipolysis, protein synthesis, and it causes increased BMR. Similarly, it has wide variety of effects on the cardiovascular system, CNS, growth, and on many other systems, which inshallah we will discuss in the next video on the functions of thyroid hormone. Then there is also non-genomic action of this hormone, and this effect occurs through second messengers such as cyclic AMP or protein kinases. The site of non-genomic action, it appears to be cell membrane, cytoplasm or mitochondria. Cell membrane, cytoplasm or mitochondria. This non-genomic action occurs within minutes, and especially it occurs in tissues like heart, pituitary and adipose tissue. Thank you for watching today's video. Stay blessed, happy learning, and Allah Hafiz.